Thanks for joining, everybody. We're gonna just take a minute or so just to let everybody filter in, and then we'll start. For those of you joining us, we're just gonna wait a minute or so to let people come in, and then we will go ahead and get started. All right, well, let's go ahead and get started here. So this is the Alcohol and Inflammation panel. And we would like to welcome you to the 2024 Symposium on Substance Use Research. My name is Joyce Bashir. I'm a professor at the University of North Carolina, Chapel Hill in the Department of Psychiatry, and I'm also the Associate Director of the Bowles Center for Alcohol Studies here. And just to tell you a little bit about my lab, we are focused on understanding neuroadaptations that occur as a consequence of stress, and how that impacts alcohol drinking and relapse. And we work in rodent models, and we're also have a line of research looking at neuroinflammatory signaling and how that promotes increases in alcohol drinking. And we're kind of bringing that all together, and now beginning to consider how neuroinflammation impacts the stress response, and how those are all tied together. So a few reminders before we begin. This session is being recorded, and we will send an email to registrants and post on social media when the recording is available. And also, this session is a webinar, so all the attendees are muted. So please use the Q&A function, and you'll see it kind of at the bottom there of your screen to submit questions, and we'll try to answer them as time allows. And I'm really excited to introduce today's speaker. We have a fantastic group of panelists here from a wide range of disciplines, as you'll see as they introduce themselves. So we'll go ahead. We have Dr. Molly Monning, we have Dr. Leon Coleman, and Dr. Tamara Phillips. And so I'd like to just take a second to do some brief introductions. So Dr. Monning, let's go ahead and if you just describe your background, your research area, and your relevance, the relevance of the work you do to this panel. Thank you.
Yeah, thank you so much. Yep, I'm Molly Monig. I'm an assistant professor at Brown University at the Center for Alcohol and Addiction Studies. And I am a clinical psychologist by training, and I'm really focused on sort of the behavioral science of alcohol and alcohol use disorder. And my work looks at the effects of both chronic and acute alcohol on inflammatory biomarkers as well as the brain. And throughout all of that, of course, I'm very interested as a behavioral scientist in how some of those biological effects might feed back and affect behavior. So how that becomes sort of a feedback loop. The methods that I use are alcohol administration in the lab. So we do controlled alcohol administration, and we measure peripheral biomarkers like chemokines, cytokines, some of the other acute phase proteins, and we also do neuroimaging. So MRI, and my primary modalities there are diffusion tensor imaging and MR spectroscopy. So I've done work in healthy adults, some work in people living with HIV, and now also looking at an aging sample. So kind of branching out from sort of, what we know about alcohol use in healthy individuals and extending that to clinical populations and other populations.
Fantastic. Thank you. And Dr. Leon Coleman.
Hi, thanks for having us today. My name is Leon Coleman. I'm assistant professor at UNC Chapel Hill in North Carolina, Bell Center for Alcohol Studies. And we have a strong interest in understanding how neuroimmune signaling and peripheral immune signaling contribute to addiction, particularly alcohol use disorder, and if we can target those pathways for new treatments. We work with animal models, with human tissue, and I'm happy to collaborate with human researchers to try and achieve that.
Great, thank you. Dr. Tamara Phillips.
Hi, I'm Tamara Phillips. I'm a professor and chair of the Behavioral Neuroscience Department at Oregon Health and Science University, and I also work within the VA Medical Center here. And I'm the director of the Portland Alcohol Research Center. And the research in my lab has been for many, many years focused on genetics. I'm a behavioral geneticist by training and interested in how genes impact behaviors. And in the case of alcohol, I'm interested in how genes impact the risk for developing an alcohol problem and how alcohol interacts with your genotype in order to impact how you respond to it. And so with regard to neuroinflammatory issues, some of our research has indicated that some of these neuroinflammatory molecules have a role both in risk and response, and whether or not their role is through their neuroinflammatory processes or their signaling processes is something that we're still investigating.
Great, thank you. So as you can all see, we've got a really great panel here, really excited to get us started. So thank you all for joining us today. We really appreciate hearing from you and having your expertise on this panel. So I guess I will kick it off with a question to get us started, but I do want to remind everybody out there, please put your questions in the Q&A feature, and we can, we'd love to hear from you all and have our panelists answer some of your questions. Okay, so to kick it off, for somebody who's kind of new to the alcohol and inflammation research, what are some of the primary relationships between inflammatory responses and alcohol use and misuse? And Dr. Phillips, if you would like to kind of get us started with this one.
Sure, I'll kick this off, and if any of you want to add things that I may miss, please feel free to do that. So I want to start by saying that alcohol is an equal opportunity toxin. It has effects on peripheral organs. It easily crosses the blood-brain barrier, and it can affect both neurons and glial cells. And for those of you who may not be so familiar, glial cells are the support cells for neurons, but they also have some of their own independent roles in the central nervous system, such as astrocytes being able to clear excess neurotransmitters or stabilize and regulate the blood-brain barrier and promote synapse formation. So alcohol can affect all of these cells in your body. And while low to moderate alcohol consumption may not cause significant inflammation, heavy drinking can lead to very serious health issues. And this can be dependent upon your individual differences, both genetics and environmental exposures, whether or not alcohol and how significantly alcohol affects inflammation in your body. It can have direct effects by increasing the production of molecules called inflammatory cytokines. These are simply molecules that trigger or increase inflammation in the body, including the brain. And while alcohol can trigger an immune response, which is your way of defending against harmful toxins or substances, it can also impair the function of immune cells. So there's a difficult situation where you're getting both increases and then impairments. This can include protecting the lungs and other organs. So, this can be very widespread. Alcohol can also damage the gut by altering the balance of good and bad bacteria. It can increase intestinal permeability, and this can lead to intestinal inflammation, sometimes referred to as leaky gut syndrome. and some of us have some expertise in this area as well. It can damage the liver. I think that's one of the most common pieces of information that people know about alcohol's effects on the body, so that the liver becomes less efficient at detoxifying harmful compounds that you might come into contact with. And this can lead to hepatitis, which is a condition characterized by inflammation, widespread inflammation of the liver. You've probably also heard of reactive oxygen species, because we're always being told to eat certain foods to deal with increases in reactive oxygen species. And alcohol, when it's metabolized to clear it from the body, can produce these reactive oxygen species, which can damage your DNA. They can damage proteins in your body. And so that's another way in which inflammation induced by alcohol creates problems, and can increase even things like risk for cancer and heart disease and other diseases. So again, alcohol really has profound effects on the entire body. Alcohol can be broken down into products called acetaldehyde and acetate. Think of acetaldehyde sort of like formaldehyde. It's not something that you want floating around in your body for very long. And those byproducts can cause inflammation if they aren't quickly removed. It can also increase uric acid, which can lead to something that you might have heard of, which is called gout, which is a type of inflammatory arthritis that causes very painful swelling and redness in the joints. So those are a lot of peripheral things. What about the brain? Chronic alcohol consumption can also clearly cause inflammation in the brain, which can lead to brain damage and cognitive dysfunction, problems with learning and memory, long-term changes in mood. And high levels of alcohol can even result in something called alcohol-induced dementia, which is very hard to distinguish from Alzheimer's disease. There's also evidence that it can accelerate the development or accentuate the symptoms of Parkinson's disease. So that's something to consider. We're learning more and more about gut, liver, brain interactions. So long-term alcohol exposure can cause immune cells actually to migrate to the brain, where they can transform into something called macrophages that contribute to inflammation and tissue damage. Macrophages are white blood cells that remove dead cells and stimulate the actions of other immune system cells. So again, you're getting increases in immune system activity. You can also get direct damage to neurons from alcohol in the central nervous system, causing them to shrink and die. And then that increases macrophage levels because they need to come in to remove those cells. It's also very important to note that if one already has an autoimmune condition or any medical condition with high levels of inflammation, alcohol can worsen the symptoms of those sorts of diseases. And so those are some of the individual differences to consider. And I want to say that some recovery from inflammation is possible from alcohol use, but that really depends upon levels and chronicity of use. And if liver inflammation progresses to cirrhosis, then permanent liver scarring has occurred and can't be reversed. So some things can be reversed and some things are not reversible. The entire brain is susceptible to alcohol-induced inflammation and damage. And there's some evidence that the frontal lobes are particularly sensitive. And this is the part of the brain that you need for executive functions and inhibitions of behavior. So effects can be really quite profound and noticeable. And the limbic system also, which is important for emotion, may also be particularly vulnerable. So that's just sort of an introduction to the number of different ways in which alcohol can produce inflammation in different organs of the body.
Thank you. That was fantastic. I really liked how you covered central and periphery, and I want to get into a little bit more about that as we go along. But first, Dr. Manning and Dr. Coleman, do you have anything that you'd like to add?
Oh, that was fantastic. I don't have anything to add. Thank you so much.
Yeah, that was very comprehensive. And my advice to anyone trying to get into it is really ask the question of why, what's your question? Why do you want to study inflammation? What does it really mean in your setting? Because it's a word we kind of throw around, but as Dr. Phillips was just describing, there are a lot of discrete cell systems and cell systems that are involved. And if you can kind of narrow it in on a cellular function or an outcome that you're really interested in, rather than just diffusely saying, well, let's measure cytokines or whatever, which can often not be helpful. If you have a real targeted focus, I'd strongly advise that.
Mm-hmm. Yeah, that's great advice. And Dr. Phillips, you also mentioned, the idea of kind of, if you have an existing inflammatory disease, let's say, that likely adding alcohol to that is not going to be a positive outcome, right? You're likely to exacerbate that inflammation. Right. I think that is really something very important to consider as we consider like human health and yeah. Mm-hmm. Mm-hmm. So that was great getting us started. And hopefully everyone out there, you got a really comprehensive idea about, this interaction between alcohol and inflammation. So Dr. Coleman, this question is going to be directed to you, and it's building a little bit more about what Dr. Phillips was saying when we consider the periphery and the central nervous system. So what is the relationship between, if you have peripheral or systemic inflammation and neuroinflammation? Okay, so that's in the periphery, and so now we're talking about neuroinflammation in the brain. And I guess after you answer that, could you tell us a little bit more how the gut microbiome could be implicated in that?
Yeah, it's a really important topic, and kind of like Dr. Phillips said, alcohol, I forget the phrase you use, but equal opportunity offender or toxicant, right? So
yeah,
it goes throughout the whole body, through all cell membranes, exerts effects on every tissue, organ tissue and organ system. So that's important to consider, that no tissue that you're studying is an island when it comes to alcohol. It's weird because alcohol has direct cellular effects, like, for example, like she said, during metabolism, but there are also cell effects. So you have what we call traditional immune cells that release factors that can regulate neurons and things like that. But then you have the central and peripheral crosstalk, which is interesting to me because it definitely happens, but you don't have to have it. So it's kind of weird. So like you can, if you're studying cell culture, using cell culture models to study neurons or microglia, other brain cell types, you can get inflammatory responses similar to those that you find in vivo, even without those influences. However, in a living organism, it obviously plays a role. So you get gut leak with heavy alcohol use, and bacterial contents or products can move into the bloodstream where they can activate immune sensors in the liver or in other organs or in the blood. And those can release cytokines that go to the brain and kind of exacerbate the direct effects of alcohol that alcohol itself has. So you have this combinatorial effect where alcohol has direct effect on brain cells, but then you have peripheral cytokines and peripheral signals that can exacerbate that. And so it's helpful to study, brain cells outside of the body, but also in the body, and make sure you're getting similar results. There's direct communication through the vagus nerve between the gut and the brain and vice versa, which can have consequences in circuits in the brain and the periphery. Also things like extracellular vesicles released from the brain or released from circulating immune cells can cross the blood-brain barrier, so you can have direct communication that way as well. Those are just a couple of ways that we know. But then lastly, as Joyce mentioned, is the gut microbiome, which has become a hot topic as of late because alcohol can cause gut dysbiosis. And several groups have been studying this, and changes to the composition of your microbiome coupled with the increased gut leak seem to have profound consequences. So you can have increased extravasation of bacterial contents that are now overrepresented or underrepresented, depending on the effect of alcohol. And also you can have metabolites that are altered from the alteration and the composition of the microbiome. And these things can promote a systemic inflammatory environment that helps drive craving. Some groups have found and other features. So it's complex because there's so many moving pieces, but you have cell autonomous effects of alcohol. You have cytokines from the periphery, like TNF, that can cross the blood-brain barrier, of extracellular vesicles, and vagus, and these bacterial metabolites. So just a lot to look at.
That is a lot. And Dr. Coleman, when you're talking about the gut microbiome, and you're talking about disturbances to it, is it possible that a treatment could be to kind of normalize the gut microbiome, and would that help, you think?
Yeah, so possibly, and a lot of groups are looking at this. I know Ali Khoshavrian at University of Chicago, he's been kind of leading in this. Yeah, and people are trying. It's a complicated order, so to speak, right? Because you have millions of bacteria and there's so many. The microbiome is so complex. It's kind of hard to narrow down to, let's target this one bacteria or this or that. But people are trying to do that. And not just only for addiction, but this could have, as kind of Dr. Phillips was alluding to, this could have implications for other diseases associated with alcohol, like cancer or neurodegeneration. So that is, I know a lot of groups are trying to do this. It's not, because the microbiome is so complex, it's not that simple, but that is a hope for a lot of groups.
Mm-hmm. Yeah, I mean, it definitely sounds, the more I learn about the gut microbiome, there's so much going on in there. So yeah.
Yeah, and I just had Bernd Schnabel, for example, as well, with alcohol-related liver disease, is making tremendous progress with this, trying to change the bacterial contents using bacteriophages and things like that. There's a lot of people working on that.
Yeah. Dr. Coleman, there's a question in the Q&A about the extracellular vesicles. So, and the question is, could the extracellular vesicles be hijacked for treatment? So could you tell us a little bit about the possibility for that?
Yeah, absolutely. So that's one of our hopes, is to be able to try and do that. So extracellular vesicles, they're multiple kinds, but they're small, for people don't know, they're small membrane enclosed structures that carry proteins, microRNAs, all kind of signals, complex signals, and they're altered with chronic alcohol use. You can potentially target them. You can remove certain vesicles using antibodies targeted against cell surface proteins on these vesicles. You can try and dialyze them, but you can also try and use supplement with protective vesicles. And so a lot of groups use like vesicles derived from mesenchymal stem cells, for example, to try and reduce some of the inflammation in certain conditions. So we're trying this. I know other groups are trying this. Consuelo Gary did some work with that. And Pascal, she's done some work on that in Italy. And so groups are trying to do that.
So is it something that could be a peripheral marker in like in clinical studies, that we could then potentially use, in our kind of our translational animal studies, like as a biomarker, or you have some kind of signature of these EVs?
Absolutely. I don't want to scoop ourselves. We're trying to do that, but a lot of people are, I think, and it's kind of it's kind of a ripe area. I hate to say it, but it's true. It's a ripe area. If Molly, for example, if you're trying to find new biomarkers, because most people don't even look in the vesicle population, so it's kind of a ripe area.
Dr. Mani, is that anything that you in your group have looked at?
We have not looked at the extracellular vesicles, no. We have looked at, I'm really fascinated by the whole process of leaky gut and the body's reaction to it. So we've looked at markers of that. And, just to link it to central nervous system, we do find that some of those biomarkers are linked to cognitive performance, for example, which you wouldn't necessarily expect, right? And whether that's mediated by neuroinflammation, I don't know, but I would posit that it probably is. And We're also really interested in you mentioned, the treatment of the gut dysbiosis. We are really interested in probiotics, as Dr. Coleman mentioned. It's hard just to say, if you have trillions of bacteria, like what, and thousands of species or however many, like what are you to target? So we're doing some like whole genome sequencing on that, just to like down to the species and strain level, trying to get like a better sort of a grip on that. And I know other groups have gotten some signal for probiotics in alcohol use disorder treatment, actually finding that it does reduce drinking more than placebo. So there does seem to really be like some proof of concept there.
That's great. Yeah, thank you both. That was very helpful. Um, let's see. So hold on. We have there's another question that just came up. So let's go through this question. Would love to hear the panel's take on the paradox of sickness behaviors in the context of alcohol addiction, namely why inflammation is linked to lower reward processing and lower motivated behavior, but we also see increased craving for alcohol under conditions of elevated inflammation. Thank you very much for that question. Would anyone on the panel like to take this question?
I can take a stab at just a little bit just to start out.
Yeah.
And I think that is actually a really brilliant question, actually, because, so like alcohol can create like a sickness type scenario, especially during withdrawal. And this could be part of the whole hyperkatifia, a negative affect contribution to driving people to drink. And if you're looking at things through that lens, you could say, well, the desire to alleviate that sickness behavior is a strong motivator. And that could be related to or an aspect of craving. And it's not necessarily full sickness behavior, but just a dysphoria, an altered affect state. So for me, and I'd love to hear other people's thoughts on this, my question really, in regard to what you stated, is really what is craving? How do you really, it's a non-descriptive word, right? It's kind of like stress. Like, what really is craving? Is it a motivated behavior? How do you really conceptualize the idea of craving? I don't know what your all's thoughts are on that.
Yeah, that's a hard thing to define, but I would also agree with you, Dr. Coleman, that there are things that override these issues that develop, right? So that craving or that desire to use can possibly, due to withdrawal effects, can override the negative consequences of inflammation or anything else. The same thing happens with methamphetamine. Talk about a toxin and extremely neuroinflammatory. But as you know, people keep using it even under very dire consequences. So there are simply things that override some of these negative sickness results of overuse.
Yeah, I mean, I see it the same way, just increased motivation to obtain and use the substance that people have their prior experience tells them will make it go away, right? So I don't really even see it as a paradox so much as, motivating that singular behavior in the context of, alcohol misuse.
Kind of
Yeah,
a competing behavior situation. Yeah.
Right, exactly.
We also we've done some studies where we've injected a neuroimmune activator and let's say LPS or imiquimod, which activates the toll-like receptor. Imiquimod does the toll-like receptor 7. And acutely it does make the animal, like if animals were trained to self-administer alcohol, and acutely it does make them ill, and let's say they're losing body weight. But then when we look out further in time, like a couple weeks later, and it's results have been inconsistent, let's say, but sometimes you will see these animals escalate their drinking, and it is kind of fascinating, like what is going on that is driving this. So is it kind of like the panel was saying, like overcoming kind of some negative aspect, and the drinking is going to perhaps dampen some of those effects. It's really thanks for the question. It's a very interesting.
Yeah, I wonder as we just discussed it, because there's the thought that other rewards are less salient, or there's less motivation for other rewards. So that could contribute, the kind of sickness phenotype could contribute to that potentially. I don't know. Loss of salience or rewarding aspects of other behaviors, but like they were saying, that drug gets linked to a relief from that, so or a reward as well. So it's really good stuff.
Strong habit.
Yeah.
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 All right. So please, attendees, feel free to put in questions. You've had some great questions, some great discussions. So I'll just come up with another question here. And this, Dr. Monig, is directed to you. What do you see are the most pressing gaps in the inflammation and substance use literature? And do you see populations that perhaps are underrepresented in these areas that we need to do better with?
Yeah, of course, that answer will really vary widely depending on field. So looking at human research, Dr. Phillips did a great job talking about like the association between chronic heavy use and all of these, disease outcomes, and how inflammation mediates those processes. That's really well established. We don't know as much about lower levels of alcohol use or the acute effects of alcohol use. So looking at moderate alcohol use, most of the population does use alcohol at below the risk levels, right? And many people think it's good for their health. I don't want to be like a total, rain on the parade. But the epidemiological research is now really kind of showing that it's not, the prior research that was showing, 2 glasses of red wine at night or whatever is good for your health are really more based on like studies that had a lot of selection bias, for example, where people who were sick quit and then they looked more unhealthy and then, in the non-drinker category, and it made that J-shaped curve. So we really do need to know more, I think, about the effects of moderate alcohol use, or like kind of low, sort of in the middle, like the low use to up to the, not the extremely heavy or chronic use that typically gets studied, I think, in humans. And then also humans, of course, we try to isolate the effects of alcohol, which is already hard enough in free range in humans. So we tend to look at the healthy populations and the young populations, right? Meaning people without other health conditions. So, most of the studies, for example, that I was looking at, just like a survey of studies that administered alcohol and then looked at the inflammatory biomarkers afterward. And all of those studies have a cutoff of like 45 or 50 in terms of age, right? So there's a huge swath of the population that is not being included at all in these studies. So I'm doing a pilot study now in ages 60 plus, trying to address that, but it really is like a big gap. And in the US population, over 50% of people have a chronic health condition, possibly more than that. So we're like thinking obesity, depression, high blood pressure, heart disease, stroke, diabetes, arthritis, et cetera. So like we are not really looking at the effects of alcohol as much in these populations with respect to inflammation. I think there's a lot of like kind of like observational research, that some alcohol use, heavy alcohol use will make those things worse. But I don't think it's been as systematically studied, the effects on the immune system in those conditions, and whether we can actually give people specific recommendations, like based on your condition, like this has been found for people with your condition. And I think that specificity matters to people when you're giving them recommendations. Otherwise, it's sort of just like, well, that doesn't apply to me. And we also don't understand very well how alcohol contributes to the development of, the common chronic diseases. So, the end organ alcohol related diseases, yes, that's been much better defined. But, like I mentioned before, you know, heart disease or dementia, things like that, like people really still do have this idea that like a couple drinks like is good for my heart, is good for my brain. And you know, the research coming out now just shows that neither of those are the case. Sadly, I'm sorry to rain on people's parade. Those earlier studies were probably biased by, you know, all these different factors. And then finally, I would just mention, as usual, people of color and women tend to be underrepresented in the human alcohol literature. And that's particularly like when it comes to treatment-seeking samples, those really tend to be predominated by, white male participants. And as, just to also circle back to aging, it tends to be more like, the treatment-seeking population tends to be more like, 40s to 50s. And again, huge, portions of the population are left out when we take that more narrow focus. So really focusing on representation of all groups in our research.
Thank you. That was great. I kind of something that you mentioned when you're focusing on this older population. I mean, those are also the older populations like have increased likelihood of disease, other diseases, right? And so to have the two there together is going to be really important to understand more about what is going on with the aging process and inflammation. And Dr. Phillips mentioned it, and then you also mentioned it. But I think that, thinking about Alzheimer's disease and alcohol and inflammation and how these can all kind of, how they're all related. Or are they all related? I mean, I think I would like if someone could maybe just talk a little bit about that. Because I think it is a really important, population that we're really learning more about, I think, and focusing research on the older population.
My short answer would be yes, I think so. And I do think it's mediated by the immune system. In addition to the direct, neurotoxic effects of alcohol in the brain. But when we see, for example, those, byproducts of leaky gut and how they stimulate different reactions in the body, we actually even see some of those microbial pathogens themselves. Actually, it was thought that they did not reach the brain, and now, in at least in animal studies, we do see some of those factors like lipopolysaccharide, which is like a bacterial component, do actually make their way to the brain in places like the hippocampus, which we know is critically impacted in Alzheimer's disease. So I definitely think that there's a relationship there. I know other folks on this panel have done work in that area.
Yeah.
It seems to me like what we really need are these longitudinal studies, right? So that you can really see how things are progressing. And it's such a huge investment to do that, especially because of the number of years we're talking about. But some of the individuals who started in adolescence, maybe, we have some hope of following them for longer periods of time and getting NIH funding to go even later into the lifespan in order to sort some of these things out. It's going to take time.
Yeah, there have been some helpful, I agree with that 100%. There have been some helpful studies as well, like in Switzerland, where they have, in the Netherlands, where they've had government healthcare for decades, and they have records. It's retrospective, but they have self-report data from people when they were younger, and they've tracked them now and later in life. And there is increased risk for dementia with heavy alcohol use, late adolescence, young adulthood. We see the same thing with animal models. And yeah, we do see if we block inflammatory signaling that it prevents the ability of alcohol to promote Alzheimer's pathology in rodent models. So I agree with you. I think there is definitely an inflammatory component, and it kind of seems like alcohol accelerates your brain, the rate of brain aging. And I think several studies, you've been using UK Biobank and things like that where that seems to be the case. And the immune system is definitely central in the whole aging process.
Yeah. We have a question. What kind of data do we have about long COVID and COVID-related disability related to inflammation and alcohol? Anyone like to talk about that?
That's a great question. I know that there is ongoing research. I believe Sarah Jo Nixon is doing some work on that. So that would be one name to look up. I'm not sure if the work is completed, but in the long COVID space, that's the work that I'm aware of. I haven't really looked at inflammation directly and alcohol related to COVID.
Yeah, I just actually saw a presentation. So there is more more people are looking at the long-term effects of kind of long COVID on the brain. And it does seem to kind of, increase some of the acute phase proteins that are seen in dementia, like tau and things like that in the brain, and enhance neurodegeneration. I don't know of anyone who's, at least preclinically, who's done it with alcohol. But there is evidence to support that one, COVID does get into the brain. It can cross the blood-brain barrier, and particularly in these long COVID settings and people with long COVID or in rodents with long COVID. Interesting, they get long COVID at similar rates that humans get it, rodent models, but those actually get long COVID. They do develop persistent neuroinflammation and these proteopathy kind of features like tau as well. So that'd be a good study to see how alcohol modulates that.
Yeah, I'm not aware of any preclinical work where people are looking at the COVID, the virus itself and alcohol response or damage. But yeah, again, like Dr. Coleman said, like you have this, you know, inflammatory response, right? And if you had perhaps a pre-existing, you know, inflammatory related disease, you're just compounding. And then how does alcohol fit into this? I think it's a really important question. Absolutely. Thank you for that question. And we are still open for questions. If anyone else out there has some questions for these amazing panelists, we would love to have them. So, can we talk about, so something that came up, I think Dr. Phillips, you said this, the importance of looking at alcohol across the lifespan. And we talked about the aging population. How about adolescents? We haven't talked a lot about adolescents, and we know that adolescents engage in a lot of binge drinking. And are adolescents, by engaging in binge drinking, driving an inflammatory state that is then perhaps going to follow them for life, right? And could someone just does anyone have any thoughts about that?
I can talk about mice from a rodent perspective. Yeah, that is one of the issues with the problems with the brain is, well, brain can have a lot of problems, but the problems with the brain as far as immune signaling goes is that one is that the cells don't turn over, right? So like your inflammatory cells like microglia, astrocytes, they don't really turn over very frequently. Now, your circulating monocytes turn over every 21 days. So if you have an immune insult in the periphery, an isolated immune insult, you may get cells that, their phenotype changes, but they'll turn over. So you won't have, for example, pro-inflammatory primed monocytes circulating in your blood forever, typically, unless you have a disease. But with the brain, your microglia, for example, they don't turn over. And so if they get a pro-inflammatory stimulus that changes them to more of a pro-inflammatory state, they'll maintain that unless there's some sort of intervention like exercise or pharmacological intervention or dietary intervention to bring them back to normal. So that's the issue with the brain. So when you have situations like in adolescence, like what we see is that you get in rodents with adolescent alcohol, binge alcohol, months later, they still have increased pro-inflammatory gene activation. And in Alzheimer's models, that correlates with Alzheimer's pathology. So it can grow and persist in the brain. And if there's no intervention, you just have that higher baseline, and that interacting with the normal age-related inflammation, it's just really not a good scenario.
Yeah, thank you. And that, I mean, and it just, when you think about, public messaging to adolescents, you're thinking about now, right? And you're not thinking about, and they're not thinking about the long-term consequences that, can accompany this high level drinking when your brain is still developing, and how that could lead to the persistent changes. Thank you. Um, let's see. And we're open for questions if anyone out there has any questions for us. One thing I would like to touch on, I guess, is stress, because well, it's my interest. But also I'm sure some of the panelists also, but it is something that Dr. Phillips mentioned when you think about environmental, we have genetics and you have environmental factors that can influence the neuroinflammation. And I think stress is something that, kind of this whole body response that can drive inflammation, and then potentially, drive escalations in alcohol drinking. And I think it's just something, kind of the link between those is very important to consider as we, and again, it's one of those things like throughout the lifespan, right? It is an important factor, environmental factor to consider within this context. And the difference between an acute stressor versus a chronic stressor, where the system is constantly kind of responding, and that can have some detrimental long-term effects. So I think that's always something that I like to think about, and thinking about, are there possible biomarkers that we can identify to potentially predict like who's going to maybe have, let's say, and these would be like inflammatory related biomarkers in terms of identifying individual differences, and perhaps who's going to show us be more susceptible to inflammation driven escalations in drinking or something like that. So it's just something that I just wanted to throw out there. But if anyone has any comments on that, happy to.
Well, the other thing about the stress axis is that's remarkable is how it sensitizes.
Yeah.
So you don't really get tolerance necessarily to impacts on the stress axis. You actually get sensitization. So smaller events can cause larger responses over time. And there's cross sensitization with alcohol. So if you have a sensitized stress axis, then you get increased effects of alcohol. Very likely even with regard to neuroinflammation. With even other drugs of abuse that have been studied in animals, these sensitized responses can be seen a year later after only a single exposure to methamphetamine, for example. So I think the stress axis is really important to consider, and I would like to see more research into exactly how sensitized it might become, or how other stressors might sensitize that system and neuroinflammation to alcohol.
Right. That's really important. Thank you, Dr. Phillips.
Yeah, there's a lot of literature that in periphery that relating or connecting stress and inflammatory signaling. So an inflammatory, insult can activate stress pathways, but prior stress can sensitize you to an inflammatory stimuli. So there's a lot of crosstalk there.
Yeah. We have another question. So in regards to gut biome and dysbiosis, I hope I said that correctly, is there any research being done on the potential for fecal transplants in addressing gut biosis related inflammation issues associated with long-term alcohol use? So going back to this idea of the periphery and the importance of the periphery. Dr. Coleman, do you have any?
I don't know. What do you think, Molly?
I've seen those studies in animals, right? And it's more proof of concept where it's either like a low alcohol use or a higher alcohol use, both through breeding. I'm not an animal research person, obviously, both through breeding and training those behaviors. And then it can go both ways. So like the fecal transplant can make from the higher drinking to the lower drinking, will make the lower drinking animal drink more. and then vice versa. So there's definitely proof of concept in the animal literature. I have not seen anything clinically, like in terms of clinical research ongoing, but there could be ongoing trials in, humans that we just haven't been published yet.
Mhm. Yeah, thank you. And I guess as we've got about 10 minutes left. So and this is to anybody on the panel, what are some important considerations in the methodology and or the measurement of inflammation in substance use research? And I guess I always think of it like, and we kind of touched on this a little bit, but how can we like leverage our preclinical animal models to have more translational potential? And is there anything we could do with, peripheral markers, for example, that we can do both in the humans and in our animals? I don't know. Does anyone have any thoughts about that?
Yeah, I mean, in terms of the markers, you mentioned peripheral, the neural markers are the hardest because we don't have a measure of neuroinflammation in humans that is direct and non-invasive, right? So we either have to wait for postmortem tissue, which great work has been done there. But if we want to look at living, breathing humans, we have some markers through positron emission tomography, which is PET. There aren't any direct measures through MRI, which is the most accessible, affordable, feasible method for, for wide scale use. So I would definitely say measures of neuroinflammation in humans that could be direct and non-invasive would be a game changer.
Yeah, the other thing is that oftentimes we because of funding issues or just practicality, we can only look at very few points in time. And I don't even mean ages, but even just, how long after a stressor are you looking at the inflammatory response? How long after alcohol administration? And it would be nicer to have more data to see how long these effects last. And also on real dosage effects. Human studies actually giving them alcohol, it's hard to do. So you are relying on self-report, and it's harder to get a real handle, I think, on how much alcohol exposure someone has had over periods of time and really accurately see what the dose-dependent effects are.
Mm-hmm. Definitely something easier to do in our preclinical animal model.
Much easier. Yeah.
Yeah. We found that, I mean, so in my lab, we've given people placebo, low dose, moderate dose, and those folks are divided into light drinkers and heavy drinkers. So you have both the acute dose that varies and then people's drinking history that varies. And both of those really matter. They really matter. So, for example, like there's not differences in the baseline for their chemokines and cytokines. We looked at an array of 5. But after we give them the alcohol, there are significant differences. And we even saw like evidence for a placebo effect, or you could call it like conditioned immune response of alcohol in 2 of the 5 biomarkers that we looked at. So that was MCP1 and TNF alpha. Those are widely used measures of, inflammation, but it was only in the heavy drinkers, not the light drinkers. So what that tells you is that, or what that tells me is that folks' drinking experience before they have whole lives before they come into the lab, and that really matters. So a lot of the studies, right? Yeah, we don't have the time or we don't have the money to really characterize what their drinking history is like or what their recent patterns are like. But I would say it's very important.
That's very interesting. Yeah.
Yeah, and going back to this idea of the sensitized response, right? Like all that drinking that they have done has changed their system, even though at baseline they may not look different. But when you throw on the alcohol, yeah, that's great. Thank you. That's a really interesting study. Can somebody or the panel just in general, does anyone have any comments on the importance of genetic and epigenetic mechanisms of alcohol use disorder?
Well, I mean, it's well known that genetic differences impact sensitivity to alcohol, how much alcohol is consumed, recovery, withdrawal, severity, all kinds of factors. So there's no reason to believe that those genetic factors wouldn't also impact inflammatory responses. I know that inflammatory molecules are definitely impacted by genetic sequence, and response to alcohol, or by selective breeding of animals. So breeding animals for high and low drinking, so that you've got these genetic effects that the only reason you would be able to breed is if you've got genetic differences. Then if you study their brains, you identify genes that are different between those animals and relate them to their alcohol drinking. And some of those turn out to be in these neuroinflammatory genes that are predictive of high and low alcohol drinking. And similarly, the effect of alcohol then on those animals with regard to the inflammatory systems tends to be different. So there are definitely those genetic differences. I've done much less in the realm of environmental factors, impacting us, giving them a stressor, looking how that might interact with those genetic differences. But genetic differences, I think it'd be really interesting though.
Yeah. Anyone else on our panel want to comment on this?
Yeah, the epigenetics is really, there's a strong epigenetic effect of alcohol and histone methylation, DNA methylation, acetylation during intoxication, and then a wave of methylation afterwards. So there's a strong epigenetic effect that directly. So we were speaking earlier about this concept of priming, both with stress sensitization, but also with pro-inflammatory gene signaling, and pro-inflammatory signaling is strongly regulated by epigenetic mechanisms. So the induction of some of these mechanisms like methylation of histones, H3K9 dimethylation, in microglia, for example, is associated with prolonged pro-inflammatory priming of those microglia. So that gives hope, I think, because I think if we can kind of normalize some of the epigenetic factors, you can't change your genetics, right? But if we can normalize some of the epigenetic factors, we could potentially normalize some of the pro-inflammatory signaling and improve some of the behavioral pathology.
And there's also some evidence that those epigenetic changes can be transgenerational. So it's really important to look at this. Yeah.
Absolutely. Okay, we'll have one final question here. What's your favorite inflammatory marker? And the one that you think doesn't get enough love in the AUD research field?
I don't know if you can ever get enough love, or too much love. But Molly, what's your favorite one?
I like MCP1, so monocyte chemoattractant protein 1, and because I think it has a lot of relevance for the gut-brain axis. It's very active in the central nervous system, and we just see a lot of correlations behaviorally, both with cognition and with craving. So I think those are two obviously really, really relevant for clinical research. So that's the one that, it does get a fair amount of love, but it's maybe not enough. There's a great study from, I think, like 2005 that shows if you knock out MCP1, it's the alcohol seeking behavior in rodents is eliminated, right? So really strong evidence for its role in, like AUD type pathology.
Yeah. Thank you. Dr. Phillips, Dr. Coleman?
Mine would be right now is interferons, so that they're really associated with sickness.